Hello.
There is an Ian Pie Day. You got to celebrate whatever you want. I choose to celebrate pie, not a number.
A very cool number.
It's a pretty cool number.
Yeah.
I agree.
Yeah, and actually, we should find the Twitter. Maybe you can find the Twitter thing. Somebody posted this beautiful thread on the history of pi, the number, and it's cool. I learned some stuff, which is neat. It's always nice. Yeah.
Pies were first made by your mama. They were delicious.
Yeah. Actually, my mom's pies were pretty good. Yeah. Your mom's pies are pretty good, too.
They're pretty good.
Yeah. Yeah. There's this blueberry one that she makes. Oh, my goodness. Like, it's not cooked blueberries. It's like raw blueberries with this cream cheese layer. Oh, my goodness.
Oh, my God. It's so juicy.
We need to ask for one of those. Yeah. Okay.
Just shout it.
Mom! Pie!
Blueberry pie, please!
Wonderful.
Hey, we should tell her to watch this.
We should. Oh.
Please like and subscribe.
To our pie needs.
Yeah.
Okay.
Previously on Piecast.
Yeah. So, today's a really fun lecture. Are these lecture class? Today's a really fun one because what we're going to do is we're going to really probe you into thinking about the scientific method, like the actual methods. And it's going to be uncomfortable. You're going to be unsure. But all we want you to do is experience that uncertainty. And we'll do it together and we'll talk about it. And I love this lecture. Okay. And it looks like you're almost done. Kyle's drawing a face. There we go. Okay. And there's some Pac-Man going on over there. This is lovely. Have we missed anything? We always miss under our video feed.
Yeah.
I think there's going to be some love for Ukraine. There is some Ukraine love, which is
Nice.
great. Wonderful. Yes. Oh my goodness, yes. Okay. Oh. Are we ready?
Yeah.
I think we're ready. Okay.
I would like a mug of that, whatever.
Of whatever pie that is,
No, yeah,
or coffee.
I like the yeah.
Yeah.
This is tea, and it's a good tea. But...
Okay. Wonderful. Okay. Here we go. I'm going to clear. Oh, folks are still coming. Welcome. Clear all drawings. Oh, do we have a screenshot? Now we do. Clear all drawings. There we go. Aw.
That's not nearly as delicious.
It's not nearly as delicious. Yeah. Okay. Oh, but first, have you seen this? This is a pretty good tweet.
This came out on the social medias last week. I think it's the end of February. So a couple weeks ago it actually happened.
With the best caption ever. Because the tweet that went out with it was nothing to do with the polar bear. Entirely to do with the audio track. It said, I don't know which family member to love more. Okay.
So this is actually not on the island. This is in the and Labrador part of Newfoundland and Labrador.
And I'm hoping...
Push the button in the...
Yeah, but will the CCs go on?
I think
Okay,
so.
we'll try it. If not, we'll stop
They're
it.
kind of sweary.
They are kind of sweary, so watch out. Harsh language.
Please be careful, Minnie. Please be careful. Oh my God. You just nose. Oh my God. Please don't hurt Minnie. Oh my God. Please don't hurt Minnie.
Get him over that house. Get out of it. Come on.
Dad, Pop, be careful.
Do I call Jeffrey?
Call Jeffrey, Katie. Call Jeffrey. Holy shit.
Oh shit! Called Jeffrey.
Holy shit.
What do you got? Isn't it pretty?
Please don't come up to the window. Auntie, keep it away from the window, Auntie. That's right, Cooper. Holy shit.
Oh, you're pretty.
Oh,
That is such a beautiful bear.
Pretty. Oh my God, Ninny, please be careful. Please be careful.
Everyone was fine.
All the dogs are fine too.
Yeah, and this, that's an amazing polar bear sighting.
Yeah. It speaks to a whole bunch of things. Like, what are bears doing right now? Well, this one's pretty healthy, but it's also, it speaks to, we've talked about in the physiology unit, the bear is kind of the lens through which we're often returning to look at ecological, evolutionary, and physiological changes driven by the climate crisis. But also just the fact that in that part of the world, they're curious.
Yep.
And they have no reason to be afraid of literally anything. So the threatening voice that you might shout at a black bear with, like, "Ah, big guy, get away!" The bear's like, "Oh,
Hello. Say that louder.
I've never eaten an ibegar, get away. I wonder if they're delicious."
Yeah.
And yeah, the dog's oddly large.
Yeah. And it's true, like, just like, you know, the crack of a gun, pointed into the air can be terrifying for a black bear. They don't affect polar bears. I've shot like right over top of the head of a polar bear to try to scare it away, and then it just looked at me, right? Because it's hearing these types of cracks all the time as it walks on the ice and moves around the Arctic landscape, and so they are really just impressive creatures.
Yeah.
Yeah, nothing like the sheep on the family farm.
No.
No.
Although my dad does talk to them in the same way. Hey, sheep!
Yeah. Yeah.
Somebody else's dad saw some in Greenland when they went there with the
Wonderful.
military.
Okay, so the population in Greenland is a little bit different in terms of their behavior. So they are a bit more wary of humans. Each human in Greenland represents more of a threat than most of the humans in northern Canada because of a different culture in terms of interactions and things like this. And in Greenland, there were sort of more deliberate intentions to sort of separate out human and polar bear populations. So they may not have been like life-threatening, but certainly there was sort of more sort of policy related to making sure that polar bears weren't near human areas. And there's a lot of humans in Greenland too, a lot of communities. So yeah, super good. Okay.
Our dad said that he was told that they would hunt down humans.
Yeah, they do. Yep. We are viable and
we're delicious.
Delicious
well,
prey.
I'm told.
Yeah, we're a little lean.
Really? That doesn't sound like us. That's not what the news tells us.
Lean, a bit gristly.
I think it actually says when it goes, "Welcome to Scotland, we're delicious." Kind of greasy.
Yeah. Okay. So yeah, we already told you what we're going to do today. We're going to practice thinking like scientists. I like this class. Okay. But first, a poll question. Let me just see if I've got it queued up in the polls. This is the same question as the homework. So hopefully you're just going to click in pretty quick because I think we gave you... No, we didn't. Okay, so we didn't give you the time scale or the biological level of organization in the homework. That was your homework, is to go in and associate. Yeah. And what we've done here is we've given you 4 incorrect ones and 1 correct one. So just go in and find the one that matches with what you've got, and hopefully it'll be correct. And I will pull up the poll while you do that. Nope. There it is.
That's a cute organ, he said upon entering the church. I had to bring it out of the gutter. The gutter.
Sweet.
Did you see Randy Rainbow's latest tweet?
No.
It's musically set to The Little Shop of Horrors, one of the films. So I've got that
nice.
The Be a Dentist song.
Oh, good.
So it's...
Cool. Just a few more seconds. Most of you have already participated. Thank you. Did you see the video that I sent you through text?
Yeah.
Yeah.
That's 2 years ago.
2 years ago. When we were
Right
so
over
much
there.
younger. Okay, wonderful.
When I had purple hair.
10 more seconds. Five, 4, 3, 2, 1.
Jackpot.
Beep, beep.
Oh. Bum, bum,
Share your
bum,
results.
bum,
Good.
bum,
Okay.
bum, bum.
Yes. The answer is E. So that's the one that is correctly matched. Arctic foxes have small ears, short muzzle. All of those things don't change. The state of those traits doesn't change over time, either in chronic or acute time periods. So you know kind of by default it's generational, evolutionary, because those differences would be like species specific or at least sort of clade specific or adaptation kind of, environment specific rather than sort of changing over time within the same individual. And it's also at the individual or the organism level, because those are talking like whole body type adaptations, multiple organ systems having to collaborate there in terms of achieving the overall effect or the overall adaptation. Cool? Great. Stop sharing.
It's a bad button. Never stop sharing.
Never stop sharing. Oh dear. Okay, we're going to throw two more terms or sort of one kind of concept at you. We're going to do this like pretty much every class. Kind of here's a thing that's part of physiology that you might need to know. And here's how it kind of fits within the big umbrella of all of the other terms.
It's like a new Lego box. So
yeah.
You've all got your big box of Lego at home, and this is like some new pieces.
Some new pieces. So we're going to talk about conformers versus regulators because it picks up where we left off asking the question about Rick Mercer versus the salmon. What are some of the differences in terms of predicting what's going to happen?
The salmon also doesn't have a TV show.
Fair enough.
Anymore.
Yeah. What's going to happen, in the context of a warming, largely terrestrial environment and also some aquatic, but that matters too, but that's for later.
I'm holding in what I want to say.
Okay. Because later. And one of the big differences, I think, from a physiological perspective for sure, is that Rick is one and the salmon is another with respect to being a conformer or a regulator when it comes to temperature, but not all variables. And so that's what we're going to parse out today, is talking a little bit about conformers versus regulators. So basically, a conformer conforms whatever variable you're talking about to meet the environmental variable. So whatever's going on in the environment with respect to the variable that you're talking about. And what am I talking about? It could be temperature, but it could also be salinity, it could be pH, it could be a whole bunch of other different things, right? Basically, it's, the internal environment of the organism is fluctuating or remaining just as stable as the external environment. And more importantly, if you're a conformer, you're not physiologically or metabolically maintaining it, okay? And a regulator is basically constantly fighting it, right? Constantly trying to maintain its own range within that variable, despite or at least maybe even in spite of what's going on in the environment, right?
Temperature, I hate you.
Yeah, in spite.
You're not my dad, temperature.
Okay, so there's going to be like this metabolic, this kind of drive from the body, from the internal body to regulate that variable.
And I think one of the amazing things, and a thread that you'll have seen kind of throughout this course and your continuing training as becoming the biologist that you already are, is the fact that there's some cases where this is just a clear, it is that or it is that.
Yeah.
And then as you unpack more and more of life whose planet they share with us, you'll find intermediates where it's like
fuzzy.
It's kind of one, but more of this with another abiotic factor. And those are some amazing bits of biology.
Yeah. Cool. The fuzzy gray areas are so interesting.
Now, many people wanted to become a vet because of fuzzy biology,
Yeah.
but really what they wanted to be was a biodiversity scientist.
Okay, so let's get our sort of classic models down so that when we do look for these strange and unusual, creatures, we know what to look for, right? So let's establish what the theory is, and then, let's celebrate the things that bust away from our theories, because they're just human-derived theories. So let's maybe take a blue line if you want. That would be fun. A blue line, and draw for me, please, using your annotation. You can do a straight line, or you can just kind of scribble it. That's totally fine. If we're going to look at a conformer, what would be the relationship between the external environment and the internal environment? And if you wouldn't mind drawing that line for me, that would be great. Cool.
Looking good.
Looking good. Yeah. Okay, so a conformer, it's going to match, right? And it's going to change as the external environment changes, right? Okay. Somebody want to pop in the chat why we don't have the internal environment on the x-axis and the external environment on the y-axis? Why is the internal environment? So why is external on the X and internal on the Y?
Porque. Dependent versus independent factor. Internal is dependent upon external.
Very good.
Internal is dependent on external. Take away the question mark.
Own it.
Yep.
Yeah, that's right. So our independent variable is always on the X. And usually always, mostly always, I'm sure somebody's broken that rule. And then on the Y we have our dependent, the thing that would depend on the independent. Thanos broke it.
Thanos, we'd plot Thanos's internal environment against the external environment.
Okay.
He caused
that.
Yeah.
Okay. Okay. So this is perfect, and that's what your line is going to look like.
Well done.
So well done, all of you. Here we go. There we go. Line of conformity. Okay. It looks something like this. Now, why have we added a little bit of nuance to this? Why is there a dotted line here?
Why have we said a dot dot dot? A dot dot dot.
Why does it not go all the way down here or all the way up here? What's going on in this space? You can also write on this. Ooh, Leah's adding to it.
There's a flagella.
Extreme, somebody says.
Extremes.
Cell may die. Super good.
It can only conform within a certain range of tolerance.
Super.
That's good.
Very good. Thanks, Leah. That's perfect. Yeah. Nice. I love that.
Ooh.
You're putting numbers to it.
You quantified it.
Super good. Okay. Yeah. It's the range of tolerance, right? So most things have a range of, all things have a range of tolerance, some very broad and some very, very narrow. Broad, I'm thinking like water bears.
Water bears, woolly bears, to go with another not a bear.
Not a bear.
But that's the brown and black caterpillars that you see in the fall.
Yeah. So they have really broad, like they can survive pretty much more than all other life.
40 to 40 kind of thing, minus the plus.
Yeah. But then other things have very narrow, and so the dotted lines really suggest that, if it gets past into the dotted area for a species, they're kind of kaput, like they're not going to survive, right? So it doesn't mean that they have these infinite ability to conform. They have to do it within a range. Super good. Yay. Okay, moving on. Now, because I realized that giving you a color for this type of graph is not really necessary, and I totally want to see what colors you pick. Let's make a rainbow. Why don't you draw for me a regulator, please? So where do you think a regulator would fall on the same axes, the external environment and the internal one?
Oh.
Pick your colors. Yeah.
Wah.
Rainbow.
Yeah.
Super. Yeah.
Oh, that's an interesting hypothesis.
What's that? Yeah, the purple. That's super good.
Yeah.
That may, over a very, very short time period, that may be exactly what it looks like, right?
Yep.
If we're integrating this over like years, then not so much.
Or on the body, maybe. If your internal environment's like core versus distal.
Nice. I like it. Yeah. Okay. And the rainbow's pretty awesome. So, good. Okay. Yes. So let's add a little bit of nuance to these lines, if you don't mind, just like we did with the conformers. Oh, it's really good though.
It is really good.
Save.
It's all saved.
Okay. There.
Yeah.
Okay. So totally right. It's a horizontal line. And again, we're dealing within these ranges, right? These thresholds for individual organisms. And so there's these kind of like theoretical kind of divots that go off from those lines. What's going on there? Like why is it not a dotted line? Why is it a blue line? And what's kind of going on those little diagonal areas?
Put it in the chat or on the slide. Inability to regulate at extremes.
Nice.
Yep.
Yep. Homeostasis can't be maintained outside of it. Adaptation. Very good. Okay, so adaptation in the physiological way.
Yeah.
Right? So it's trying, it's doing its best to acclimate, but it ain't happening, right? Like it's eventually, if we push it to the extremes for a longer period of time, then we're going to get into the dotted line area of being kaput, right? But there is some ability to try to, in some cases, it's mostly just endure.
Yeah.
Right? Until whatever stressor it is in the environment stops, right? So if we're talking about a cold night, you may make it if you shiver for a bit and you do your very best and then if it starts to warm up, you can then go back down to your, normal, line of happy homeostasis. So there is usually this kind of like emergency, trying to adjust phase, after which point, if the stress lasts for a long period of time, then it's game over.
2 catch-up questions. Just reiterating the difference between conformer and regulator. So these are strategies kind of cartooned as extremes where for some abiotic stress, that X variable, so the external environment with regard to temperature or salinity, for example, the organism tracks whatever the environment does. Versus a conformer, did I just reverse these?
Yes.
I did. So the regulator maintained. That's what you're looking at. That is the slide. So that's the homeostasis. So that's us with regards to temperature, for example. Then the conformer is an organism whose internal environment is the temperature of the environment in which it sits.
Yeah. So imagine you just stop heating your apartment or your house. And eventually that apartment or that house is going to start to conform with the external temperature, right? It's going to probably, if you're in Guelph, it'll be bloody hot in the summer, and it'll be frickin' cold in the winter.
It's never cold here.
Yeah.
Yeah. And then there was another one. Oh, the difference between adaptation and acclimation.
Previous class. Adaptation we use in evolution. Acclimation we use in physiology. Okay. So, this is great. We're going to clear.
And continue.
So hyporegulation happens around the line of conformity. So in this area, there would be the attempt by the physiological system or the organism to hyperregulate. And here we're getting into hyporegulation. Okay, usually. Don't worry too much about that stuff. But what we want to ask then is, what kinds of animals are conformers? And this is going to bug Smith, because when we talk about kinds of things, he gets really specific.
We'll talk about
Watch me
control myself.
Open the paraphyletic groups. So can an animal be both a regulator and a conformer? And this is really what we want you to kind of focus on. Okay. So let's take a look at this table, and what I'd love is if we could work through this table. So we can try to do this together. It would be really great. I'd appreciate it. So if you want to get a stamp ready, how about you get a green check mark ready? And no, sorry. A red heart. We'll do temperature first for all the things. Thanks, Emma. I'm totally just going to change it on you now. Red heart. Temperature, please. For mammals, are they conformers or regulators?
Just like Hey Rosetta. We were listening to their show, their song, Red Heart.
That's right.
Oh, there's a fight.
There's a fight. Good. Okay. For birds, are they conformers or regulators? Go ahead. Thank you. Super. Reptiles and amphibians, are they conformers or regulators? Fishes, conformers or regulators? And then this is where we'll see the little vein in your forehead. Frog.
I don't have a vein.
Invertebrates, are they conformers or regulators? Oh, do you see what they're doing?
You make me cry.
You're amazing, all of you. And also, feel free to populate the conformer part, too. That's fine.
Don't bully. Do not bully. Do not force conforming.
You're amazing, all of you. Well done. Okay.
Because I'm going to have to play the other side of the table and say, well, yeah, mostly.
Mostly. They are mostly conformers, but they're in the invertebrate world because it's so big.
It's like all of things, right?
All of things. There are tons of exceptions, right? Okay.
And you know about them.
Yeah.
They make your honey.
Oh,
Bees make heat.
They
Bees are thermal regulators. Many animals, the woolly bear is a thermal regulator. There's all sorts of subtle things that you can do that aren't as they're maybe not as intricate as ours for thermal regulation, but they are thermoregulatory morphological morphologies and behaviors.
Yeah. Naz is writing something. I'm not sure we're going to get it, but I'll
says,
try.
Oh.
Something. Is that a G maybe? I don't know. Okay. We'll watch you. Great. Okay. Let's do ions. So ions, just think about,
That was fun.
that was great. Think about salinity. Okay. And let's use, I think it's a blue arrow. We'll do blue, if you don't mind, pick your blue arrow and let's fill out this table for salinity. Now, whether mammals are, regulators or conformers. Go ahead. And then birds. I don't know what that is. Okay. And then reptiles and amphibians. And the fishes. Look at that. And the invertebrates.
Interesting.
Very interesting.
Yeah.
Cool.
That's interesting.
Yeah? Why?
Well, we need to take our class to BC.
Why?
To look at rivers and to go upstream where the salinity goes down and then goes downstream where the salinity is higher.
Yeah.
And then look at what happens to how things are spread out.
Yeah.
Both the fishes, all the invertebrates.
And the things that spend time in both water systems, right? Like salmon are just ridiculous physiologically in terms of what happens to their bodies.
Well, there's support. So we'll have a class trip.
Oh, wonderful.
We'll have a poll. Would you like to go to BC? Yes or no?
Okay. So good. Yeah. Thank you for this. This is great.
Sorry, just a redirection.
Yeah.
What is meant by salinity/ions? So we are meaning, I'm talking. We are talking about like the salt concentration. How saline, if you're in a brackish water, very salty water versus kind of farther and farther upstream along the river away from the ocean, you get more and more fresh water, less and less salt in your water. Just as a simple example of a gradient, of an ion gradient or a salinity gradient.
Yep. And then some of us have kind of salty bodies and most of us have freshwater bodies, right? Yeah.
But there's a salinity gradient in terrestrial environments too.
Yeah.
I'm sure you've seen videos of those parrots in the Amazon flocking to clay salt licks because of the salinity that they are not getting because they are so far away from the coast.
That's right. So we filled it out, and I think you did a better job. But here we go. Here's kind of what our thoughts were. And there's all sorts of question marks down here because it's just a monster paraphyletic kind of group called the invertebrates, which is, yeah, not cool of me to have done. But it would have made this table really, really long. And
we could do it by file. We could. That's interesting.
Yeah.
We could do like a 40 column.
Yeah.
I'd be into that.
Let's do that on a Saturday night.
Ecdysozoans. Ecdysozoans. What
Okay. Great. Wonderful. So we're going to talk about exceptions and things like this because those things are really kind of fun to talk about. But for the rest of this class, let's kind of play around with the scientific method and again practicing interpreting graphs and stuff like this. Okay, so we'll probably have time for a couple of mentees as well. If you want to open up Menti, you can go to www.menti.com and we'll have the instructions on the slide when we get there, but just if you want to do that now, that's totally fine. Let's talk about winter flounder. And
which is how we feel around February.
Yeah, and they are equally as exciting as freshwater mussels in the Great Lakes. They're amazing, I think, because, and like many fish, so this is not particular to them, but like many fish that live in really cold water, it's kind of physiologically interesting and a little freaky because the body of a fish is freshwater, right? You know that whole thing about most of us, we're all 70% water. Well, that water is largely fresh water, right? There's very little salt in a body compared to, say, the saltwater environment. But salt water doesn't freeze solid until about -2 degrees Celsius. And fresh water starts to freeze at 4 degrees Celsius and then freezes solid or can freeze solid at 0 degrees Celsius. So if you have a largely freshwater body in your body fluids, in salt water that could be colder than the freezing point, you may have a bit of an ice cube problem, right? So why don't winter flounder freeze? Is the question. It's kind of a good question. And I've experienced Antarctic water. It's freaking cold. It's colder than the Arctic water, but you wouldn't be able to really tell the difference. It's like a matter of a couple of like 0.02 or whatever. And these guys, these winter flounders, are in absolutely frigid water, but they don't freeze, even though their frozen point, the freezing point, is associated with a freshwater system. Okay? So let's explore this. We have an observation. These things don't freeze solid. And we want to start exploring some of the hypotheses that could explain that. And you're all working now on writing hypotheses and things like this. It's something that you've already done in the seminars. But we want to kind of show you how it unfolds, and then, of course, the nuances and the complexity associated with writing these, and then writing predictions and things like this. So that's what we're going to do. We're gonna think about some hypotheses. We're going to make some predictions. We're going to test out these predictions all in the next 15 minutes. Okay, so observation. Why don't fish freeze, right? And imagine there is a little bit of literature out there already, some research that's been done. So this is a really good way to ground some of your hypotheses, okay? So let's imagine that there is this available information already. What we'd love for you to do is to take a few seconds, go to www.menti.com, and we're always going to ask you, share 2 conclusions, 2 things that you can conclude from these data. go. Super. Well done. Okay. Keep on going. Wonderful. Thank you. Keep going, keep going. It's really good practice. I hope, anyway. If you don't mind it. Okay. Let's take a look. Okay. Inversely related. Yeah, inversely proportional for sure. As antifreeze proteins go up, or proteins, the concentration goes up. The two are inversely related. There's a normal distribution in one of them. That's really good, but an inverse normal in the other, so also normal. There we go. Lowest in the summer months, highest in the summer months. Seasonal acclimatization occurs. That's really nice. Fish have antifreeze proteins. Antifreeze protein concentration increase in the colder months. Antifreeze increases during colder months. Freezing point increases during warmer months. Excellent. Really good. Here, lower freezing point is warm enough in summer. Excellent. Good. Good, good, good. Okay. Yeah. And so that's like a real thing. These are real data. It's a real trend that we see in winter flounder. Here, the amount of antifreeze changes by temperature. So the different times of the year, the fish are less prone to freezing. Awesome. Love it. It's a totally testable hypothesis for what comes next, right? Here's an observation. Now, we had an observation that fish don't freeze. That's a curiosity observation. Then we had an experiment that looked at antifreeze protein concentrations and the plasma freezing point. Now we have a whole bunch of new testable hypotheses that you've already put forward, which is fantastic. So imagine then that we're going to take a look at what the trigger is, right? Looking at these data, what triggers this change in the antifreeze protein concentration? And an antifreeze protein is a protein that's made within the body to change the freezing point of a particular bodily fluid, right? So it'll decrease the freezing point, even though that fluid should be freezing, it isn't, okay? So what's the trigger? And your hypotheses really do speak to the trigger, right? When you're talking about the cold months and the summer months, the warm months, and the temperature and all of these things. So let's set up a little bit of an experiment to figure out how temperature affects the upregulation or the production of antifreeze proteins. Here we go. So we have the antifreeze proteins increase, right, in the fall and decrease in the spring. That's our secondary observation. Our secondary question is, what is the trigger? And our hypothesis could be, in cold water marine fish, the trigger for synthesis for antifreeze proteins is, and if we go back to some of your hypotheses, right, some of your comments about the conclusions, see we have here, the amount of antifreeze proteins changes by temperature. It's a perfectly reasonable hypothesis that we can go ahead and test, okay? So let's test it. So the trigger is temperature. If the hypothesis is true, then we should see something, right? But first, before we even get to the prediction, what type of methods could we use? What could we do, actually, in order to figure out whether temperature is the trigger for the antifreeze protein? So what would you do? Like, you have a budget, say, let's just say unlimited. I know.
The best budget.
It's ideal. What would you do? And so it's another Menti that says, what methods? What would you actually do? Like with fish. I assume you'd use fish. What would you do?
Some people might use in silico methods. Sample some flounder during different seasons, it says in the chat. That's an idea.
That's an idea. Nice. You're doing well. Good.
I see some good ideas in the chat too.
Oh, good.
I'm not reading
Okay.
them out until
we give you a minute.
Yeah.
Because it's not obvious. Like, there's no one way to test a hypothesis, right? And that, to me, is where a lot of creativity can come in. There are some, historically, like, astounding and elegant experiments that have been done by very creative people. And then the ones that I really like are the scrappy ones, like the the pulling stuff together kind of, to try to make it work kind of ones, right? Where like the problem solving ones. I like those a lot. And just in terms of methods, I think it's a largely overlooked part of science, and I really like thinking about it. Okay. You have some really good ideas. So let's take a look here. Fish in the freezer. Yes, you can do that. Although if you don't put it in water in the freezer, then you may be adding a confounding variable.
just making supper.
Yeah.
For later.
Super good. Move them to a different temperature. Yes, right? You absolutely have to do this. This is like bare minimum something that you need to do, because otherwise, if you're just sampling them in the wild, you don't know what the trigger is, and you're just going to be recreating that previous graph, right? Okay, so take fish and change temperature during the same month. Brilliant. Yeah. Measure antifreeze concentration. Exactly. How many fish? How many fish do you want to do this to? Change temperature for somebody, yeah, or write on the screen, whatever. How many fish? Recreate temp artificially to rule out if it's a seasonal pattern. Very good. Groups. How many groups of fish in different bodies of water at different temperatures? This is nice. This is different, right? So this one here is about taking the same fish and changing the temperature. This one is about taking a bunch of fish and putting them in different temperatures. Which one is... No, neither of them are better. I'm not even going to give you a trick question. But what are the pros and the cons of that, right? Of this one versus this one? What are the advantages and the disadvantages of that design, right? Are you going to put all of the fish in the same tank in your group? Or are you going to put one fish in its own tank by itself, right? Whole bunch of other pros and cons associated with that. More fish, more data. I love that. Super good. Put the fish in multiple different climates at a time and see how they react over a period of time. So let's go all the way down here. Measure concentrations in 1,000 fish. Oh. Could you imagine if we had 1,000 fish? Alter temperature of pond and remeasure. This is in the wild. You're going to go and change the temperature of a pond. That's genius. Then you don't have to worry about, controlling all the variables because it's all in the wild. I love it. Impossible to do, but delightful that you thought of like an ideal. Set up habitats that mimic their natural habitats. Exactly. Use different samples of flounder at different water temps. This is great. Yeah. Place some fish in warm water for a longish, I love longish, that's great, period of time, and some others in cold water for a period of time, then check the protein levels. Really good. Okay. So there isn't just one way to study the temperature of a trigger, to study the existence of a trigger. And you've come up with all sorts of different ways. We've probed a little bit to try to get you to refine and think about it, right? There's all sorts of different ways of setting up an experiment to answer a question, which is often why no one experiment is going to allow you to fully answer your question. Because of the pros and the cons of each of the different designs, right? But imagine that we set up one where we have a control group of 20 fish that are going to be maintained at 10 degrees Celsius. with an experimental group of 20 fish, and we're going to decrease the temperature slowly until we get to below freezing point of fresh water. Okay, so we're going to do this over time. Then we're going to measure the plasma antifreeze proteins, and we'll see whether or not, or we'll see what happens. Okay, that's the experimental design. So let's draw some predictions then. So imagine we're the control fish and we are at 10 degrees Celsius the entire time. What would you predict would happen to their antifreeze protein concentration if temperature is the trigger? So let's do a blue line, please. Just show me what would happen in the control fish over time. So feel free to, there, Amarashkan's taking the lead. Yeah. Good, 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 wonderful. Yeah, right? If temperature is the trigger and there is no temperature change, we wouldn't expect to see antifreeze protein concentration changing. Cool? Okay, let's draw the experimental line then. Go ahead. Don't worry too much about like, is it exactly here or there, but just what are we looking there, Nathaniel? Good. Good, good, good. Excellent. Okay. So we do this all the time in our labs. Not every lab does this, but for both of us, we really like our students to come up with prediction graphs, because then when you collect your data and you make a graph, you just line it up and go, oh, it's this one. Right? It's so much easier that way than to try to figure out what's going on. So we always insist on prediction graphs before we even start sampling.
And it's a really helpful way for you to present your data. So it's helpful for you to go back and see, what have I actually found? But then it's helpful for you when you're telling your data's story as well.
That's right. So the experiment was done, and this is what happened.
Huh?
Now,
what the flounder?
What the flounder? So this is a WTF moment, right? Because this was not supposed to happen. And it did. And this can cause quite a bit of anxiety in a scientist until you start thinking about it scientifically, right? So is temperature the trigger then? If this is your graph, is temperature the trigger? What do you do?
So, first of all, you check your thermometer.
Yeah. Check your methods.
And then you talk to your technicians. And
That's right.
Or yourself.
That's
did
right.
I actually do what I thought I did? And
Yeah.
you did.
Yeah.
It's none of those really not very interesting things.
That's right. Always check your methods, right? Maybe there was something that was contaminated. Maybe there was a problem with some of the machines you were running.
Yeah.
Always check your methods. And then imagine this is a totally
It's
well
there.
done experiment. It is still there. So no, big no's. And there was a little no over here that I really liked too, because it was gentle, but this one is like loud shouting, no! Okay, so what do we do next? We pack up and we give up as a scientist.
Take up that music career.
Wonderful. No, we get curious, right? And we start to ask a little bit of questions, right? So let's go back to here and to originally to the hypotheses that you had given with our two conclusions, right? If it's not temperature that's the trigger, what could the trigger be?
Hey, Douglas. You're late to class.
What could the trigger be? Nice. Time of year. Good. What?
Time of year appears in the chat as well. Season,
Yep.
circadian rhythm.
Nice.
Circadian
Nice.
rhythm.
Yeah. So how does a flounder know what time of year it is?
Google Calendar.
How does a flounder know what time of year it is? What might be the actual trigger, the actual thing?
Internal clock, mating season, day versus night.
Lovely.
Oh, light
Good.
cycles.
Yes. Good. All of these
Sunlight.
things can be tested, right? I love the internal clock idea.
Yeah.
I love the mating season. Like if it's a hormonal clock, that's
Yeah.
freaking genius. Yeah. Day and night. Yeah. All of these can be tested, right?
Yeah.
And all of them would lead to these horizontal lines except for one thing. And that's your trigger, right? So let's skip to the kind of end. We're running out of time anyway. But let's go and take a look. So here is photoperiod. Here's your experimental design for photoperiod. So photoperiod is the relationship between the number of hours in the day that are daylight and the number of hours in the day that are not. And that changes with the seasons, right? The days get longer and the days get shorter. So we're going to have 20 in your control, and this time instead of controlling temperature, we're going to control light with 12 hours and 12 hours. And then in your experimental groups, we're going to have 20 fish on different light regimes, right? So different light from 10 to 14 all the way to 8 or 6 in light and 18 in darkness, okay? And now we're going to measure antifreeze proteins. And based on this graph, is photoperiod the trigger? Yeah. Lily says yes. Good. Everyone says yes. Cool. Yeah, because it changes, right? It changes. So independent of temperature. Think about that. That's messed up, right? Why would photoperiod evolve to be a trigger for an antifreeze protein if it's actually an adaptation to changing temperature? And with that, we will say goodbye so that you can think about it, and we'll take it up next time. Awesome.
See you Wednesday. Be good to yourself. Be good to others.
Bye. I know. Just keep waving.